Hello friends, welcome to my channel Easy Life Science. Here I am talking about enzyme inhibition part 2, irreversible inhibition, allosteric inhibition, suicide inhibition and end product inhibition. So please subscribe my channel Easy Life Science, like my video, share it to your friends and don't forget to press the bell icon so you will get the notification of my newly uploaded video. So let's start. Irreversible inhibition. In this type of the inhibition, inhibitor molecule binds to a functional group of active sites of the enzyme by a strong bond such as covalent bond. And so because of this binding, the inhibitor molecule cannot dissociate from the enzyme. Thus, it permanently destroys the catalytic property of the enzyme. The functional group of the active site are hydroxyl group, sulfhydryl group and amino group. Irreversible inhibitor is a non-specific and cause a dead end of the enzyme activity. The inhibitor can bind to a free enzyme or enzyme substrate complex and destroy the activity of enzyme permanently. For example, iodoacetaamide. This bind with a sulfhydryl group of an enzyme and inactivate the enzyme permanently. The enzyme activity after a irreversible inhibition does not get regained on a dialysis or by increasing the substrate concentration. We seen in a competitive inhibition, the substrate molecule and a inhibitor molecule compete to each other for binding to an active site of the enzyme. By increasing a substrate concentration, we can restore or regain the enzyme activity. Unlike this, here we cannot regain or restore the enzyme activity. Here the enzyme activity completely lost. A variety of poisons such as iodoacetate or reactive oxygen bases are the irreversible inhibitors. In terms of the kinetics of the irreversible inhibitors are similar to that of the non-competitive inhibition. We seen in a non-competitive inhibition, the substrate molecule binds with an active site of the enzyme. This is an active site of the enzyme to which the substrate molecule get bind. The enzyme has another site known as allosteric site to which the inhibitor can bind, and because of the binding of inhibitor, the kinetics of the reaction get changed, and this is known as a non-competitive inhibition. In a non-competitive inhibition, there is a decreasing of a Vmax value and no change in a Km value. In same manner, the irreversible inhibition decreases a Vmax value but no change in a Km. In this graph, if we plot a graph against a substrate concentration on a x-axis and a velocity of a reaction on a y-axis represented by a v and substrate concentration represented by a k. There are two lines when an inhibitor molecule is present shown in a pink color and no inhibitor, that is a black color. When there is an inhibitor, you can see for the Vmax get decreased at time of a presence of an inhibitor and no change in a Km. These are the kinetics. When irreversible inhibitor molecule is there, value of a Vmax get decreased and no change in a Km, and this is similar to a non-competitive inhibition. Some examples are listed here. Irreversible inhibitors and the enzyme inhibited by them and their therapeutic use. The first example, disulfiram. The disulfiram, it is a medication used to treat the chronic alcoholism by producing an acute sensitivity to ethanol. Disulfiram works by inhibiting the enzyme acetaldehyde dehydrogenase or aldehyde dehydrogenase. And this enzyme is a hepatic enzyme of a oxidative pathway of alcohol metabolism. This enzyme converts acetaldehyde to a acetate. Disulfiram irreversibly inhibits aldehyde dehydrogenase by competing with a nicotinamide adenine dinucleotide, that is a NAD, at the cysteine residue in the active site of the enzyme acetaldehyde dehydrogenase. So this inhibits the activity of this enzyme aldehyde dehydrogenase, so it is useful for treatment of alcoholism. Then the next example, cyanide. Cyanide inhibits the cytochrome C oxidase activity. Cytochrome C oxidase is the terminal complex in a electron transport chain, or it is also known as a complex fourth in a ATC. Cyanide, it is an inhibitor. This inhibits the cytochrome C oxidase by combining with iron within the enzyme, which results in a prevention of a transport of electrons from cytochrome C oxidase to the oxygen, and thus this cannot generate a ATP and cell get die. So the cyanide inhibits the activity of enzyme cytochrome oxidase and it inhibits the ETC. Then fluoride. The fluoride ions inhibit the alcoholic fermentation and glycolysis. Fluoride strongly inhibit the enzyme enolase. The enzyme enolase, which convert a phosphoenol pyruvate to a pyruvic acid, and this enzyme is important in a glycolysis. In presence of inorganic phosphate, fluorophosphate ions are formed, which when bound to a Mg ions forms a complex with enolase and inactivates the enzyme enolase. So the fluoride irreversibly inhibit the enzyme enolase, so that they inhibit a EMP pathway or a glycolysis. Then the malathion. The malathion is an organophosphorus. It is used in an insecticide. This compound and other compound like sarin, soman and a tabun specifically inhibit the activity of enzyme acetylcholinesterase. Malathion exerts its action on the nervous system of insects by irreversibly inhibiting the activity of acetylcholinesterase enzyme, thereby allowing acetylcholine to accumulate at a cholinergic synapses and enhancing cholinergic receptor stimulation. So this malathion inhibit the enzyme acetylcholinesterase and useful in a organophosphorus insecticide. Then the next example, diisopropyl fluorophosphate, that is a DFP. It is a very potent neurotoxin. It combines with the amino acid serine at the active site of the enzyme serine proteases and acetylcholinesterase. These enzymes that deactivate the neurotransmitter acetylcholine. DFP, that is a diisopropyl fluorophosphate, is a surrogate of a nerve agent sarin. It is an organophosphorus compound which inhibits a neuronal enzyme acetylcholinesterase, so it is useful in a nerve gas because it inhibits the enzyme activity of enzymes like serine proteases and ACE, acetylcholinesterase. The next example, BAL, British anti-leukocyte or dimercaprol. It is a medication used to treat acute poisoning by heavy metals like arsenic, mercury, gold and lead. Dimercaprol is a dithiol that acts by forming a stable 5-membered ring between its sulfahydryl groups and certain heavy metals. the ball, thereby neutralizing the toxicity of heavy metals and eliminate the heavy metals. So the ball or dimer caprol, it is useful for antidote for heavy metal poisoning. Then the next suicide inhibition, or it is also known as mechanism based inactivation. It is a specialized form of irreversible inhibition, also known as a mechanism based inactivation, in which inhibitor molecule makes a use of enzyme's own reaction mechanism to inactivate it. The inhibitor molecule, which is structurally analogous to a substrate, is converted to a more effective inhibitor with the help of an enzyme to be inhibited. Means, in short, we can say that in this suicide inhibition, the enzyme literally commits for a suicide. They utilize a normal enzyme reaction mechanism to inactivate the enzyme. Some examples are listed here: drugs, their products, and a target enzyme, and they're used in a therapeutic purpose. A first example of the inhibitor or a suicide inhibitor, that is allopurinol. The allopurinol inhibits the xanthine oxidase activity. It is used to prevent or lower the excess of uric acid in a blood. The allopurinol is a purine analog and it is an isomer of hypoxanthine. This inhibitor allopurinol works by reducing the production of uric acid in the body. High level of uric acid may cause a gout, arthritis, or a kidney stone. To lower it, the drugs allopurinol is used so that it inhibits the activity of enzyme xanthine oxidase, so no uric acid is saturated and we get saved or protected from a gout or a kidney stone. The next example, 5-fluorouracil. The 5-fluorouracil, 5-FU, is a effective chemotherapeutic drugs developed as an inhibitor of thymidylate synthetase, which leads to thymine-less cell death, and it is also a pyrimidine analog misincorporated into a RNA and a DNA in place of uracil and a thymine. The 5-FU, that is a 5-fluorouracil, blocks an enzyme which converts the cytosine nucleotide into the deoxy derivative. It is a medication used against a cancer. It stops the cell from making the DNA and may kill the cancer cell. Then the aspirin. The aspirin and other such drug or other such non-steroid anti-inflammatory drugs, NSAID, can blocks the activity of a COX, that is a cyclooxygenase enzyme. The COX or a cyclooxygenase, the enzyme which leads to formation of a prostaglandins, and this causes inflammation, swelling, pain, and a fever. In other words, the aspirin suppresses the production of prostaglandins and thromboxanes, because the aspirin irreversibly inhibits the activity of a cyclooxygenase by acetylation of a serine residue in a active site of the cyclooxygenase. So the aspirin and such molecules are required for a prostaglandin and thromboxane synthesis inhibition or a suppression. Then the next, difluoromethylornithine, DFMO, difluoromethylornithine or e-fluorinethine, and is sold under a brand name, that is a Fanica. This drug or inhibitor is sold under a brand name Fanica, DFMO, difluoromethylornithine, and this inhibitor can bind with a covalent bond irreversibly to the enzyme ornithine decarboxylase. It is a medication used to treat apricot trypanosomiasis and excess hair growth on the face of human. Specifically, it is used for second stage of sleeping sickness caused by a trypanosoma, and this drug DFMO inhibits the activity of enzyme ornithine decarboxylase. So these are some examples of suicide inhibiting drugs. The next, allosteric inhibition. The allosteric inhibition, an enzyme has a site where the substrate can bind, known as an active site, and rather than their active site, there is another site known as a allosteric site to which the inhibitor molecule can bind and inhibit the activity of an enzyme. Some enzyme possess an additional site other than the active site known as allosteric site. They are unique sites on enzyme molecules. Allosteric effectors, the substance bind at allosteric site and regulate the enzyme activity. It may be positive or a negative activators or effective. When because of a binding of a effector, enzyme activity is increased, then they are known as a positive allosteric effectors. When the enzyme activity is decreased, then the effectors are known as a negative allosteric effectors. If we plot a graph at time of presence of allosteric inhibitor, we got a sigmoidal curve. In this allosteric inhibition, the inhibitor molecule is not actually looking like a substrate. That means it is not a substrate analog. And this inhibition is partially reversible. Like a competitive inhibition, when the excess of substrate is added, it gets reversed. In this case, or in terms of a kinetics, at time of allosteric inhibition, value of Km get increased and the value of Hmax get decreased. When the inhibitor molecule binds to a allosteric site, the configuration of a catalytic site is modified so that substrate cannot bind properly. It will get clearly in this graph. This is a line at time of presence of a allosteric inhibitor. It is a sigmoidal curve, and this is a line at time of presence of a substrate or at time of absence of inhibitor. In this case, at time of presence of allosteric inhibitor, the value of a Km get increased and the value of a Hmax get decreased. So these are the kinetics at time of presence of allosteric inhibitor. As said earlier, the allosteric inhibitor or the molecule may be positively activator or a negatively activators. Suppose this is an enzyme, and this enzyme has a active site to which the substrate can bind, and this is a allosteric site. When the inhibitor molecule binds with the allosteric site, it modified the active site in such a way that there is no further binding of a substrate. So no ES is formed and no product is formed, and this inhibition is known as a negative allosteric inhibition. In other case, the inhibitor molecule binds to the allosteric site and change a configuration of a active site in such a way that the substrate increase its ability to bind active site. It means that the substrate can fit to the active site perfectly and ES complex formed and product is formed, and this is known as a positive allosteric activation. have a look on some examples of allosteric inhibitors. The glycolysis, first example, in a glycolysis, the enzyme phosphofructokinase PFK, the enzyme that carry out synthesis of fructose 1,6 diphosphate from substrate fructose 6 phosphate. The PFK is allosterically activated by a high level of adenosine monophosphate. So here the AMP is the activator of PFK. The PFK enzyme, phosphofructokinase enzyme, allosterically inhibited by high level of ATP and a citrate. So the ATP and a citrate, they are acting as an inhibitor. Therefore, the high amount of AMP can reverse the inhibitory action of ATP. Therefore, the activity of enzyme increases when the cellular ATP to AMP ratio is lowered. Then the next example, in a TC cycle, isocitrate dehydrogenase. This enzyme is inhibited by the ATP and activated by a ADP. Third example, the enzyme glycogen phosphorylase. Glycogen phosphorylase is one of the phosphorylase enzyme, and this enzyme catalyzes the rate limiting step in glycogenolysis in animals. Enzyme releases glucose 1 phosphate from the terminal alpha 1,4 glycosidic bonds of a glycogen. The enzyme glycogen phosphorylase is inhibited by ATP and activated by AMP. The next example, the acetyl coenzyme A carboxylase, that is ACC, acetyl coenzyme A carboxylase, that is ACC, is a biotin dependent enzyme that catalyzes the irreversible carboxylation of acetyl coenzyme A to produce malonyl coenzyme A. ACC enzyme is multi-subunit enzyme in most prokaryotes and in the chloroplast of most plants and algae. The most important function of ACC enzyme is to provide malonyl coenzyme A. This is a substrate for biosynthesis of fatty acids. So the activity of this ACC can be inhibited by AMP and activated by a citrate. In same manner, the enzyme fructose 1,6 bisphosphate and pyruvate carboxylase. The fructose 1,6 bisphosphate, the enzyme catalyzes the conversion of fructose 1,6 bisphosphate to a fructose 6 phosphate in a gluconeogenesis and a Calvin cycle, and which both are the anabolic process. So the activity of these two enzymes, fructose 1,6 bisphosphate and pyruvate carboxylase, is inhibited by a AMP and activated by a ATP and citrate. So the fructose 6 phosphate, this is a substrate for PFK, that is a phosphofructokinase. When this phosphofructokinase acts on a substrate, the molecule or a product, that is a fructose 1,6 bisphosphate, is formed. This enzyme, yellow colored, is an enzyme, and this enzyme has two allosteric sites. One is for ATP binding and another for AMP binding, and it has one active site to which the substrate can bind and fit. So this is a structure of the PFK enzyme. When the ATP molecule binds to this allosteric site, it modified the active site in such a way that the substrate cannot bind further. That means it inhibits the activity of an enzyme. But when the AMP binds to the allotteric site, then it modifies the active site in such a way that the fructose 6 phosphate, the substrate, fit easily into the active site of the enzyme, and product, that is fructose 1,6 bisphosphate, is formed. So next type, negative feedback inhibition or end product inhibition. Negative feedback inhibition or end product inhibition. The number of catabolic pathway, including EMP pathway or a TC cycle. Lot of enzymes are necessary where the A substrate get converted into a product B through the action of enzyme first. The B reactant get converted into a product C through the activity of enzyme second. But the final product in this pathway, suppose this is a D and this is a product, and this D product can negatively give a feedback and stops the catalytic activity of this enzyme first, so that no more B and no more C products are formed. This is known as a end product inhibition or a negative feedback inhibition. This is a specialized type of allosteric enzyme inhibition required to control the metabolic pathway for efficient cellular functions. End product of metabolic reactions produced in excess inhibit the first or a regulatory enzyme in the sequence. The end products are controlling their own rate of production. Some examples of negative feedback inhibition or end product inhibition are the enzyme ALA synthase. The enzyme ALA synthase is inhibited by the product heme. The ALA means 5-amino-levolinic acid synthase. 5-amino-levolinic acid synthase. This enzyme synthesizes a heme in a liver and erythroid cells. ALA is a allosteric enzyme and its regulation is due to the feedback inhibition by heme itself. Heme is a final product of the pathway. Then next, cholesterol. The cholesterol synthesis is by a pathway known as a mevalonate pathway. The substrate 3-hydroxy-3-methyl-glutaryl CoA. This is a substrate for enzyme HMG-CoA reductase, and this HMG-CoA reductase, this enzyme is blocked or inhibited by the final product or the end product cholesterol, because the HMG pathway or a mevalonate pathway, in this pathway the cholesterol is an end product and this inhibits the activity of the enzyme HMG-CoA reductase. The next example, glucose 6 phosphate, and this is an inhibitor of enzyme hexokinase. The hexokinase is an isoform of a glucokinase. The product of its reaction, glucose 6 phosphate, does not inhibit a glucokinase but inhibit the hexokinase. This is a most important enzyme in a glycolysis. It leads to formation of a glucose 6 phosphate from a glucose. After a formation of glucose 6 phosphate, the product, it inhibits the activity of hexokinase. So from this point, no EMP pathway continues. So it is a end product inhibition or a negative feedback inhibition in a glycolysis. Then acyl coenzyme A, this product gives a negative feedback for inhibition of enzyme acyl coenzyme A carboxylase, that is ACC, which is necessary for the fatty acid synthesis. Then the enzyme aspartate transcarbamylase, or it is also known as aspartate transcarbamyl transferase or ATKase. This catalyzes the first step in a pyrimidine biosynthetic pathway. Cytidine triphosphate, that is a CTP, is an allosteric and end product inhibitor of ATKase, aspartate transcarbamylase. So it is a final product in a pyrimidine synthesis and blocks the activity of enzyme aspartate transcarbamylase. So these are some examples of end product inhibitors. So here in this image or in a chart, I can summarize all the inhibitions: competitive, non-competitive, irreversible, allosteric, suicide and feedback. Looking one by one, in a competitive inhibition, the inhibitor molecule is structurally similar with the substrate and it competes for binding to the active site to the enzyme. It competes with the substrate and activity of such enzyme get regained or restored by increasing the substrate concentration. In a non-competitive inhibition, there is no any structural similarity between the inhibitor and a substrate. In irreversible inhibition, the inhibitor binds tightly to the enzyme through a covalent bond and does not fall down from an enzyme, and totally or permanently the enzyme activity get destroyed. Then allosteric inhibition, in this case the inhibitor molecule binds to a allosteric site of the enzyme. Suicide inhibition, the inhibitor molecule makes use of enzyme's own reaction mechanism to inactivate it. In feedback inhibition, the end product or the final products of the pathway inhibit the earlier enzyme of a metabolic pathway. In a competitive inhibition, the substrate compete with an inhibitor. In a non-competitive, there is no any competition in between substrate and inhibitor. In a irreversible inhibition, the inhibitor molecule can carry out the conformation change in an active site of the enzyme. The competitive inhibition can be restored by increasing the substrate concentration. So about the kinetics, in a competitive inhibition, when in a reaction there is the presence of competitive inhibitor, the value of a Km get increased and no change in a Vmax. In case of presence of non-competitive inhibitor, no change in a Km but Vmax get decreased. In irreversible inhibition, same that of the non-competitive, no change in a Km but Vmax get decreased. At time of a presence of allosteric inhibitor, Km get increased, Vmax get decreased. And in the last column or a row, the examples are there. You can take the screenshot of it because the multiple times on this same chart questions are asked for a set life science exam, NET and to the GATE exam. So it is very important, take the screenshot of it. So importance of the enzyme inhibition. The enzyme inhibition is useful for understanding the regulation of enzyme activity within living cells. It is also useful for elucidating the cellular metabolic pathway for causing accumulation of intermediates. It is also useful for identification of catalytic and functional groups of the active site of the enzyme. It gives information about substrate specificity of the enzyme. Also useful for study the mechanism of catalytic activity. The enzyme inhibitors have a therapeutic applications. They can be used as a drugs and in a medicine. So this is about the enzyme inhibition. If you like my video, subscribe my channel Easy Life Science, share it to your friends, and don't forget to press the bell icon so you'll get the notification of my newly uploaded video. Thank you once again.